we'll get started. Thank you. I'm Kelly. I'm one of your TAs this semester. So today we'll be going through some case studies. The idea here is to, there we go. So the fundamental principles you're learning in this course, we want to help you apply those to broader concepts, some real-life situations. So we'll be presenting some case studies today. There'll be 3 of us coming up. And what we ask of you is to investigate the questions that we raise in each case study. And these questions may appear on a final exam, so it is testable material. You'll notice some of the concepts that we're going to cover have not yet been covered in lecture, but they will be. So you can always refer back to these case studies as you learn more about the adaptive immune system. Throughout the rest of the semester, we will go through them in tutorial with your TA, and the slides from today will be posted on Avenue. All right. So I will present the first case on Crohn's disease. And first, I have a video for you on a case report of someone with the disease.
He studies calculus, plays video games. He runs cross-country for his high school team. Austin Clark is a typical American teen. He studies calculus, plays video.
I never know when it's just going to hit, because sometimes it's just out of the blue, random.
The hit Austin is referring to Crohn's disease, a chronic inflammatory condition of the bowel. He studies calculus, plays video games. He runs cross-country for his high school team. Austin Clark is a typical American teen, except for one thing.
I never know when it's just going to hit, because sometimes it's just out of the blue, random.
The hit Austin is referring to Crohn's disease, a chronic inflammatory condition of the bowels that affects perhaps 700,000 Americans.
The most common symptom is at some point you will develop a change in bowel habits.
In most patients, that means frequent diarrhea accompanied by abdominal pain and even the passage of blood. The symptoms, as Austin Clark knows, can be life-altering.
It's embarrassing just having to get up and go to the bathroom all the time, and then people are just like, oh, he's going to the bathroom again.
Dr. Vijay Yajnik co-directs the Massachusetts General Hospital Crohn's and Colitis Center. He says that while doctors have a better understanding these days of Crohn's disease, they still don't know what exactly causes it.
There are parallels to autoimmunity, but it's not an autoimmune disease. It's a chronic inflammatory disease.
There is no cure for Crohn's disease, but Dr. Yajnik says the symptoms can be managed using medications, and the sooner treatment begins, the better.
We know if you treat early enough, we can really control the disease. Patients are in complete remission, not just somewhat better. They're actually completely normal.
Some of the more effective drugs used in Crohn's disease are known as biologics. They are given by intravenous infusion about every 6 weeks.
Quality of life improvements are dramatic, and the healing of the intestine is dramatic.
Austin Clark is one of those Crohn's patients using a biologic drug. He says it works to a point.
After 4 or 5 weeks, it ends up to wear off, and then I have that week where I'm not doing so well.
During that week of not doing so well, Austin would like his peers and family to remember this.
I have a disease. I can't control it, and there's no cure. So I just have to deal with that as best as I can.
In Marlborough, Massachusetts, Jim Morelli for CNN.
So to recap some of the main features of that case, so it was Austin Clark. He's been diagnosed with Crohn's, and he's just a teenager, so the disease has an early onset generally. He suffers from abdominal pain, diarrhea, as well as bloody stool. And he states that he never knows when the symptoms of Crohn's are going to hit, and sometimes it can kind of come out of the blue, so it's not expected. To treat his Crohn's, he receives intravenous infusions of a biological drug, and this helps him control the disease, but as he stated, it's not a cure. So a little on the background of Crohn's. It is a type of inflammatory bowel disease. It also falls under the umbrella with ulcerative colitis. What happens is individuals will experience chronic inflammation of the gastrointestinal tract, and this most commonly occurs at the terminal ileum and or the colon. However, the inflammation can be anywhere along the gastrointestinal tract. Crohn's disease has a tendency to be chronic with patterns of relapse and remission. However, it's never cured. And the etiology is currently unknown, but it is speculated that it's a complex interactions of 4 main variables. So luminal microbial antigens and adjuvants that may be present, as well as the immune response to these microbes. Some environmental triggers may play a role, and the genetic susceptibility of the patient. So I'll touch on genetic susceptibility and the immune response a little bit more. If we look broadly at the immune response in the gut, you have an interplay between both innate and adaptive immune mechanisms. So you'll recognize some cells secreting antimicrobial proteins, which you know to be a part of the innate immune system. As well, you can see some IgA production from plasma cells. And near the lamina propria, you also have some tissue-resident cells, for example, dendritic cells, which will constantly be sampling antigens from the luminal side and presenting those antigens to T cells. So my first question is that you are to investigate would be how is an imbalance in T regulatory cells and T effector cells contribute to the pathogenesis of Crohn's disease? As well with Crohn's disease, there's a microbial dysbiosis that's observed. So this can occur when you have a disequilibrium between symbiotic commensal microorganisms and also potentially harmful or pathogenic ones. So the next question in relation to this would be how is a balanced gut microbiome critical to prevent infection with C. difficile, which is a normal member of the gut microflora? In looking at genetic susceptibility, to date there are 4 major genes associated with the development of Crohn's. Up to 22% of individuals diagnosed with Crohn's, they also have a first-degree relative with Crohn's, so there's a heavy familiar component to it. And most of these genetic variations that increase your risk are also involved in the regulation of the immune system. So one of these mutations has to do with NOD2. So the next question to look into would be how can a NOD2 mutation confer an increased risk of Crohn's disease? In terms of treatments, there's a couple different options that doctors can pursue. The first would be corticosteroids. So these will suppress an inflammatory immune response. However, they're not recommended for long-term treatment due to adverse effects that can occur. Some individuals will also be recommended to have a surgical resection of the diseased area of the bowel. However, in approximately 50% of patients who undergo this procedure, Crohn's will return in a different section of the GI tract. So as these are imperfect treatments, there's also been new developments in regards to monoclonal antibody treatments. So you haven't learned too much about antibodies thus far. So I'll go over a little bit here. A monoclonal antibody would be specific to one epitope, and these antibodies can have various mechanisms of action. So they can interact with soluble or cell surface proteins or various other molecules, leading to antagonism. Monoclonal antibodies can also induce cell signaling, They can activate complement pathways and also facilitate direct cytotoxicity. So one of the monoclonal antibodies involved in the treatment of Crohn's is infliximab. So I would like for you to look at the target of infliximab and how that may be used to treat Crohn's. A newer monoclonal antibody treatment called vedolizumab targets the alpha-4 beta-7 integrin. So it blocks this. And what you'll have to do here is look at where alpha-4 beta-7 is expressed and how this blockade may prevent or reduce intestinal inflammation associated with Crohn's. So those are all the questions I have for you in regards to this case study. They're all listed here, but again, these slides will be posted. And that's it, so I'll hand you over to the next one. Thank you.
I'll hold on to it. So today I'll be talking about HIV, human immunodeficiency virus, and I want to cover 3 broad topics that are pretty much really heated right now and are constantly discussed. So I'll begin by giving a little background. Sorry. So HIV is a lentivirus, which means that it's a class of virus that when it infects its target cell, it uses reverse transcriptase to incorporate itself or change its RNA into a DNA and ultimately incorporates itself into the host genome. It can stay latent, so it could be transcribed actively. And the structure is the RNA is in the middle, right? And it's surrounded by GB120 proteins. So the GB120 is pretty much essential for infection into the target cell, which is in humans typically CD4 T cells. And primarily the reason why CD4 T cells are the primary target is because they express CD4, which is one target, and once the GB20 binds to the CD4 T cell receptor, it also binds to the CCR5, which is also almost exclusively present on CD4 T cells and not any other cell. So this is just a broad overview of the schematic of a transmission. So typically, you have to get the HIV virus pass through the intrinsic barrier or the epithelial layer, right? This could be in the mucosal tissues most often, right, because it's the most common way of transmission. But the rates of HIV transmission is actually surprisingly really low because this layer is playing a really good job at keeping the virus out. So in a vaginal transmission, you have 1 in 200 virons passing through. So only 1 in 200 of these could penetrate through that. And only in a case where this barrier is disrupted in some way, it could be hormonal influence, could be mechanical damage, could be anything, only when that the barrier is disrupted could the virus go through. But once it goes through, it initially starts with your innate cells and taking up that virus, doing what they usually do, right? And they would go to the draining lymph nodes and activate the adaptive immune response in a few days, in a span of a few days. But what happens is the targets for the HIV virons are the CD4 T cells. So they would infect it and pretty much lead to they would replicate in these cells and disseminate throughout, right? And ultimately what happens is you have both actively replicating virus and those that stay latent and are there. So the CD4 T cells pretty much serve as a reservoir for the HIV virus. So the reason that HIV has been such a big problem is because it mutates frequently, right? Whenever you get that immune response going, whenever you get that specific T cell and the B cell response going, the virus changes. So those cells are no longer able to target those, because by the time you get those T cells, the targets are not there anymore. They changed, right? So you have a small portion that could be cleared by your CD8 T cells, right? But you have a resistant strain, which are no longer the targets of the CD8 T cells, and they escape that attack from the CD8 T cells. And this is why you have such a complex repertoire of HIV virus. So initially what happens, as I said, is you get two forms of infection. One that would undergo constant replication and dissemination and lead to cell death. But you have a portion of those infected cells staying latent, not being actively, the virus is not actively transcribed and translated. So they're the ones, these are the ones that cannot be targeted by antiretroviral therapy, the drugs that are used to treat the HIV infection. Because antiretroviral therapy pretty much targets the replication, the virus that are actively replicating, right? And in these cells, they're not replicating, so these cells are untouched. That's why when you're on those therapies, right, you still are not HIV cured. You're not cured from HIV, because if you stop the treatment, your HIV dose or the viral titers goes back up. So HIV, as we know, HIV targets the CD4 T cells. And where are the CD4 T cells present in large numbers? The gut mucosal tissues, the lymphoid organs, and the lymphoid tissues, right? The lymph nodes, spleen, where the CD4 T cells are present in high numbers. So pretty much, if you look at the histopathology later on, you would expect to see no CD4 T cells down the road because of the infection. So this graph shows the CD4 T cell numbers throughout the years, right? So initially, you get infected, and then the virus is replicating in your CD4 T cells, and your CD4 T cell count starts to drop. And then you get the spike back up. This is because of your homeostasis efforts. The bone marrow tries to replenish those CD4 T cells, and you get that spike. But then it gets exhausted, it can no longer take it, and it starts to decline. So at this step, right, you're constantly replenishing, and it's being constantly destroyed. And over time, it gets to a point that it no longer can be replenished. Your bone marrow and your immune system is pretty much exhausted. So what I would like to ask you guys is, what are the consequences of CD4 T cell deficiency in the context of a respiratory infection or in the context of vaccination? Because as we know, vaccination or viral infection or any other sorts of infection, T cells, specifically CD4 T cells, play a key role, as you will learn in later lectures as well. The other question that I would like to ask is the role that the CCR5 plays, right? So as we know, CCR5 has to be present for HIV infection to occur, right? And so CCR5 is a chemokine receptor, just like the receptor that you guys studied last week, or that was on your midterm for CXCL8 or IL-8, right, neutrophil. So if you have that absent, or if that receptor is mutated, right, the person would be resistant to HIV infection. But although this is beneficial in the context of HIV, there has to be consequences in the context of other infections, right? So the question is, provide one plausible consequence of CCR5's mutation or absence in the context of an influenza infection. Another topic that's constantly discussed is the route of transmission, right? The most common route of transmission for HIV is sexual intercourse, right? So during anal intercourse, it's the GI tract, right, the anus and the rectum. And during vaginal intercourse, it's the vagina and other components of the female genital tract, right? And a number of studies have indicated that the rate of transmission is different between the two barriers, right? Anal sex has a greater portion of transmission, or a greater rate of transmission. And this is pretty much could be explained by the immunological principles that are behind this, right? So the question here is, what contributes to the differences in the transmission rates that you see between the GI tract and the genital tract? And to finalize and give you a clinical example, right, we have Mr. X, who's 48 years old and is married and has a daughter, right? 6 months ago, he was admitted to hospital for fever, weight loss, and a hand infection. The blood work showed decreased blood lymphocytes, right, or very low blood lymphocytes. They tested him for HIV, and he was positive. So the doctor prescribed antiretroviral therapy, right? Keep in mind that the wife and the child tested negative for HIV. So the questions are, which cytokine release during an HIV infection leads to weight loss? And more importantly, provide one plausible explanation for why the mother and the child tested negative for HIV. So the answer to this has to be in an immunological concept. It can't be that the guy was cheating on his wife or stuff like that. So yes, keep that in mind. Yes, with that, I'll end. Thank you.
All right, everyone. Can you hear me? Carl? Yeah? Cool. All right, so I'm going to be talking to you about the immunomodulation behind Staphylococcus aureus infection. Now, Staphylococcus aureus, a lot of the things in immunology, we can kind of derive their function based on the derivative of their words. So Staphylococcus is actually, I think, Greek for a bunch of grapes. And as you can see here, under a scanning electron microscope, you can see that these bacteria actually cluster together kind of like a cluster of grapes. And the aureus part of the name actually comes from its beta hemolytic activity, which means that when plated on a blood agar plate, you can actually get this clearance of red blood cells, and you get this typical golden color on the plate. Now, it's a gram-positive organism, and its infections can have a wide variety of pathogenesis in someone, whether it's just a commensal colonization where the person doesn't have any outright pathogenesis, to more localized infections such as abscesses filled with pus, or even systemic infections such as toxic shock syndrome. Now, Staph aureus can be spread from person to person very easily through direct contact or by sharing things like towels, razors, bedding. And for that reason, it's one of the most common hospital-acquired infections every year in the United States. It's a very efficient colonizer of humans. As I mentioned, 15 to 30% of people are actually persistently colonized in the upper respiratory tract, but this organism is also native to the urogenital tract in women as well as the gut microbiota. So one of the questions that I want to propose to you guys is, how is it able to colonize, let's say, our nostrils, despite our body's intrinsic defenses? We learned about this previously. We know that our body has all these epithelial barriers and these intrinsic mechanisms to keep bacteria away. So what are some of the mechanisms that Staph aureus could have to circumvent these mechanisms and allow itself to colonize us? Now, extracellular bacterial infections, such as from Staph aureus, the hallmark of a staph infection is sort of this abscess filled with pus. Here you can see it a little bit better. It kind of just looks like a pimple or an ingrown hair. So that's called a boil, whereas if you get something which is called a carbuncle, it's basically a bunch of these very close to each other, but that whole area gets inflamed and filled with pus. And this is very typical of a local staph infection. But this brings to question, what innate immune cell is the most abundant circulating cell that is rapidly recruited to sites of extracellular bacterial infections? And what is the relation of this cell to the pus seen in these abscesses? So as I mentioned, staph is able to cause a wide variety of pathogenic effects in people, whether it's boils in the skin. In the lungs, it can actually cause pneumonia. In the heart, it can infect valves, particularly in people who are getting valve transplants or in intravenous drug users. It can grow on the valve there, and that's then called the vegetation. It can also cause urogenital tract infections, as well as systemic infections, such as toxic shock syndrome. And so, as you can see here, it can have local effects as well as systemic effects throughout the body. And the reason why staph is able to do this is that it produces a wide variety of virulence factors depending on the phase of growth of the staph bug. So during the initial exponential growth phase, this is when staph is initially colonizing you. It produces a wide variety of surface-bound proteins, such as protein A, which allow it to help colonize and help avoid early immune detection. Whereas later on in its growth, in this stationary phase, it's able to make these secreted proteins, particularly TSST1, or toxic shock syndrome toxin 1, which can have more systemic effects on the individual, allowing for dissemination of infection. Now, upon initial Staph aureus infection, you can have B cells that produce antibodies that can recognize these molecular patterns on this bacteria. Bound antibodies leave their Fc region exposed, so they can then act as opsonins and increase phagocytic activity. So these Fc regions are recognized by Fc receptor molecules, which can be found on antigen-presenting cells and phagocytic cells, such as macrophages and neutrophils. And these receptors can bind the Fc region of the antibodies on these bacteria, leading to opsonization and increased efficiency of phagocytosis. But as I mentioned previously, Staph aureus can produce this staphylococcal protein A, or SPA, and what this protein does is it actually sequesters the Fc region of antibodies, preventing them from binding. So this brings to light what are the consequences of this SPA protein on phagocytosis, and are these bacteria still able to be phagocytosed even with this Fc region sequestered away? If these bacteria do manage to become taken up by phagocytic cells, Staph aureus has also developed some proteins that it releases to deal with some of these antimicrobial components within the phagolysosome. And this is where I'm going to leave it up to you to do a little bit more research. But how does Staph aureus escape destruction within the phagolysosome, and what antibacterial mechanisms are impaired as compared to which ones remain intact to deal with the bacteria? Finally, as I mentioned, in the stationary phase, which is later on in terms of Staph aureus infection, it is able to secrete these exotoxins. In this case, we're going to talk about this bacterial superantigen, TSST1, which was mentioned previously in Dr. Larcher's lecture. The purpose of a superantigen, as you can see here, is that it can interject and interrupt this MHC class II TCR binding recognition, so that you don't need to have specific binding with this peptide. It actually has this sequence here, which disrupts the TCR-MHC interaction and can cause non-specific T-cell activation. And what this does is it actually helps Staph aureus avoid antigen-specific adaptive immune responses. So toxic shock syndrome toxin 1, or TSST1, how this differs from a conventional antigen. A conventional antigen, when bound to its cognate MHC-TCR interaction, will only activate 1 in around 10,000 T cells and antigen-presenting cells when presented on an MHC class II molecule. However, TSST1, as I mentioned, can interrupt this binding and cause nonspecific activation of 2 to 20% of all of your CD4 T cells in a nonspecific manner. So if you're getting this nonspecific activation of 20% of your T cells instead of 1 in 10,000 of your T cells, what are some of the consequences you can get from that? This activation leads to both the APC and the T cell generating massive amounts of cytokine secretion. But what are some of the downstream effects of systemic inflammation due to oversecretion of these pro-inflammatory cytokines? All right, that's it. I'd like to thank you all and have a good day.